Hi, thanks for coming. I'm extremely honored to introduce today's seminar speaker, Dr. Eric Betzig, the group leader of HHMI G-Linear Research Campus, who pioneered new optical imaging technology for biology. Pushing the boundary of optical resolution, Dr. Betzig has made possible real-time observation of subcellular structures well beyond the diffraction limit for visible light. For this revolutionary accomplishment, he shared the 2014 Nobel Prize in Chemistry. After graduating from Caltech in physics, Dr. Betzig continued his graduate study at Cornell University and became a PI in Bell Lab. There, he refined the near-field optical microscope, an early form of super-resolution microscopy, and became the first to image single fluorescence molecules under ambient conditions, while determining their positions to more than 0.2 micrometers. For this accomplishment, he received the William O. Baker Award for Initiatives in Research, which was at the time the National Academy of Science Award for Initiative in Research. Despite this accomplishment, Dr. Betzig was frustrated with the physical limitation of near-field microscopy, as well as academic community, and left research. Following time raising children, he worked in his father's machine tool company and developed a very cool technology that's kind of having the sort of energy principle you find in hybrid cars. However, it was not commercially successful. Only 2 devices were sold after millions of dollars spent developing this technology. In 2002, Dr. Betzig left the position, which may be the darkest and hardest time of his life, with age 42, unemployed, and not sure what to do next. However, there was a light. Inspired by Dr. Michael Davidson's work on photoactivable proteins, he hit upon the solution to make the microscope to work, the super-resolved microscopy technology. With the help from his best friend, Dr. Harry Hess, he privately built the microscope in the living room of Dr. Hess' house and observed subcellular structure at the nanoscale resolution, basically seeing single molecules in action in living cells using optical microscopy. However, Dr. Eric never rested on his invention. Within months receiving Nobel Prize, Dr. Betzig reported a new exciting imaging technology, lattice sheet microscopy, which enabled researchers to see how the cell truly divide and how the embryo develop in real time. Now Dr. Betzig tackling more exciting bioimaging technology to open new windows into molecular, cellular, and neurobiology. Ultimately, his goal is to try to combine all those technology to be able to look deep in a multicellular context, non-invasively and fast, with methods like lattice sheet, and then bring the super-resolution technology to add the high spatial resolution on top of that. Dr. Eric Betzig demonstrates perfectly what it means to be look beyond the boundary when exploring scientific frontiers. His unique experience also demonstrates the scientific spirit a true scientist and innovator should be. He always appreciates the value of hard work, and he told me that you don't have to be smart, but you have to work hard. And he followed his passions and take risks. So in closing his Nobel lecture, he said, people are always exhorted to take risks, and that's fine. But it's not a risk unless you fail most of the time. To all the unknown people out there who have gambled their fortunes, their careers, and their reputations to take a risk, but in the end failed. Remember, it's the struggle to self that's its own reward, and the satisfaction that you knew that you gave everything you had to make the world a better place. So it is an honor to welcome Dr. Eric Betzig. Please join me. Welcome.
Okay, well, thanks, Lynn, again today for that, and thanks again for the opportunity to be here. I've been quite inspired by all of the different types of biology I've seen over the last couple days here, and I really look forward to seeing what you guys can do with advanced imaging tools in the future. Now, I want to make an apology to anybody who came to my talk yesterday, because there is a little bit of overlap, and that means you have to hear the same bad jokes twice. And so, I have these down. It's just like a regular stand-up routine over and over again. So here we go. So, in my mind as a physicist, one of the most intriguing questions in science is how we go from single molecules, which are inanimate, and for protein molecules expressed stochastically inside of the cell. And somehow these darn things come together, self-assemble, to create the animate cell, which can move, eat, excrete, reproduce, and is actually robust under external perturbation, despite that what's happening at the smallest level is happening stochastically. How does that happen? Well, electron microscopy and other high-resolution tools are great, but in my opinion, the only way you're going to understand an animate object is by looking at it doing its thing live. And that's why you have to use non-invasive visible light. The problem, though, is that if that's the size of a protein molecule, then that fuzzy blob is what it looks like in a regular optical microscope. So we're 2 orders of magnitude away from where we need to be in terms of the resolution of the microscope to see the dance of those single molecules inside of the cell. And so that's kind of the problem which has motivated much of my career. Again, there was a false start when I was doing super resolution in grad school in Bell, but the idea that finally worked fairly well was that if you look at that fuzzy blob, I think you can all agree that despite its size, you can find its center to much better precision than the diameter of the blob. The problem is that in a normal biological sample, there's so many molecules packed close together that their fuzzy blobs overlap, and then you can't find the centers at all. And so the key was when my buddy Harold Hess and I took a trip to Florida State and learned about a new type of fluorescent molecule called photoactivated GFP. Now, in a normal fluorescent molecule, you shine blue light on it and it glows green. But in a photoactivated one, if you do that, nothing happens. Unless you first shine violet light on it. Then you activate it, and then blue light makes it glow green like that. And so we realized immediately on hearing about this that this would be a way out of this problem, because we could turn the violet lights down so low that just by chance only a couple molecules will come on at a time, and then likely they'd be separated from one another enough that you could find the centers of their fuzzy balls, plot those positions, turn on a new subset of molecules again and again and again. If you sum up the fuzzy blobs, you get basically back to the diffraction limited image. But if instead you plot where their centers were, you start to build up a much higher resolution image. So that's called photoactivated localization microscopy, or PALM. So when we had this idea, we were both unemployed and casting around for an idea. And this idea both excited us and terrified us at the same time, because we were like, this is so ridiculously easy, why hasn't anybody done this? So we knew we would have to work fast, because otherwise we'd be scooped, because somebody's got to be thinking. We were right. There were other groups right on our heels. And so it was going to take too long to write a grant or get VC funding or something else. So we said, screw it. So we put $25K each of our own money in it and built it in Harold's living room, because he wasn't married, so there was nobody to complain. And then very quickly, we went in 6 months from the idea of PALM And a key aspect of this was hooking up with Jennifer Lippincott-Schwartz at NIH, who with George Patterson had developed PAGFP. And we were able to cart this thing from here to Bethesda. And like I said, within 6 months, we went from these types of images of a cut through lysosomes to this, or to better appreciate the resolution from that to that. So the beauty of PALM is that, now there's all sorts of problems that I'm going to get into later, but the beauty is that you can get 20 nanometer resolution in your living room. It's that simple. It's really nothing more than the trick of how you label the sample, but beyond that, it's a wide field microscope, a sensitive camera, and a little bit of software, and that's it. Okay, so it's in principle very simple. So 2005 was the luckiest year of my life because not only did PALM fall in our laps, I had never heard of Howard Hughes Medical Institute in 2004, but by 2005, I was on their payroll in their new Genelia Research Campus. And so Harold and I went from rags to riches overnight. And they hired us without even knowing about PALM, so we were very lucky. But we started to do, for the next couple of years, this was sort of the gold rush years of super resolution, where everybody is staking out turf. And so we did a number of applications. With my postdoc, Hari, we were able to show the proteins that look co-localized inside of focal adhesions, the points of contact of the cell to the extracellular matrix, are actually not co-localized when you look at them at high enough resolution. With Jan Lipphardt's group at Berkeley, we were able to show that chemotaxis proteins, which form these clusters inside of the bacterium, form a distribution of sizes and positions which are actually predictable in terms of a stochastic model of self-assembly and doesn't need to evoke active transport to get this arrangement. With Tom Blandpeet's group at University of Maryland, by doing single particle tracking PALM in live dendritic neurons, we were able to show in these spines that, that the actin is polymerized only in certain discrete locations. Harold then built the most amazing PALM microscope ever, where he used two microscope objectives opposed to one another to interfere the light emitted from each molecule with itself in order to get extremely high axial resolution as well as transverse resolution. And with Claire Waterman's group was then able to deduce the architecture of all of the proteins in these focal adhesion contacts from the extracellular matrix up to the cytoskeleton. And in a few years later, in a paper that he did with Jennifer's group, was able to show that the escort protein, which normally is shown in EM images and scrolls below viruses, actually invades inside of the virus itself. And so that's all the good news of PALM, but there are lots and lots of pieces of bad news for all forms of super resolution. The first problem is that generally it takes a long time to get a super resolution image. And so people don't look at live cells most of the time, but fixed ones. But chemical fixatives, as you can see here, drastically can change the ultrastructure that you're trying to observe. So this is looking at actin filaments which get blown out and so forth and rearranged when they're fixed. This is looking at the endoplasmic reticulum, which gets all aggregated, as you can see here, when you go from live imaging to fixed imaging. And so I go to a lot of conferences where the theme of the conference is seeing is believing, and I go up to the podium and I say, how can we believe what we see? And this is, as a physicist, this is something that really disturbs me in imaging science and biology, or really almost anything we learn in biology, because the system is so sensitive to perturbation that I just am concerned that we hardly ever see the cell in its physiological state in almost anything we do, and this is something that we're going to come back to again and again in this talk. So, but the bigger problem, other than just the fact that you may be changing the ultrastructure, is the fact that you're looking at one time point of what's going on. And so when I was getting really frustrated with the limitations of PALM in 2008, I was very motivated by a talk I heard by Scott Fraser at Caltech, where he said, if the goal of biology is to understand the rules of the game by which molecules can create life, then just as with the football game, there's a limited amount you're going to get with any super high resolution technique, because it's going to be like if I had perfect snapshots of a football game In one snapshot, the quarterback is going back to throw a pass. Next one, the fullback is hitting the hole. The next one, the cheerleaders are making a pyramid on the sideline. And you're wondering, how are these events related to one another? But if you can just watch the game or watch a movie of the game, then it's much easier to deduce the rules. And when I heard that talk, I said, shit, I've been looking at dead stuff all my life, and it's finally time to start looking at live cells. So that's what I wanted to do. And there is a way to do that with PALM, and it's called SPT-PALM, where you photoactivate subsets of molecules in live cells and then track their diffusion or their active transport thereafter. In this case, with two proteins, one which is very mobile in the membrane and one which is not and forms these virus-like particles in this case. So a modern example of using this was my collaborator James Liu at Janelia, who used this to study the Huntington protein in Huntington's disease. These are the nuclei of two live cells, this with the wild-type protein, this with the mutant which creates the disease. And you can see that this protein forms tiny little aggregates that come and go, but generally is sort of monomeric in there. Whereas here, with the glutamine repeat, they form these very large aggregates. And James was able to show that these aggregates act like traps for these transcription factors that get kind of stuck and slowed down in there, which impedes their search on the DNA and hence downregulates gene expression. And so basically you're seeing at the single molecule level a type of event which then can have downstream phenotypic consequences leading to a disease. So the problem with SPT-PALM is, again, you're taking snapshots of subsets of molecules. And the biggest problem with super-resolution fluorescence is that fluorescent molecules are an indirect reporter of the thing you care about, which is the protein to which they're putatively attached. So there's a lot of problems in terms of the length of the linker that's connecting it, but a bigger problem is just having enough goddamn label in there to decorate densely enough these tiny little structures you want to see. So for example, if I take a pattern like this and I start stochastically adding to this ideal sample fluorophores with a probability based on the brightness there, if I go sort of 2 per period, which is what's known as the Nyquist limit, which is the theoretical limit, it's still not enough, and you have to go up 10 times beyond that. If you translate that into what is happening in a real cell, it means you have to have incredibly high density of label, typically much higher than what biologists are used to be able to get to that point. It's not that you can't do it, it's that it's damn difficult to do, and you have to worry about all sorts of things like overexpression artifacts and so forth as you're doing that. But the bigger problem that you face is just fairly obvious, I think. If I want to have higher spatial resolution, that means my image has to have more pixels in the end. If it has more pixels, that means I'm taking more measurements. If I'm taking more measurements, it's going to take more time. And it also means I'm throwing more potentially damaging light at the sample for a longer period of time. So if you're going to be honest, there are always trade-offs between spatial resolution, damage to the cell, how fast you can go, and how deep you can image. And the history of super resolution is about a bunch of people with myopia who are trying to get as far as they can to the tip of that pyramid and not realizing all of everything else that they're giving up in the process. So the one guy who understood these trade-offs much faster and earlier than all the rest of us was Mats Gustafsson, who developed as one of the pioneers of another super resolution technique called structural illumination microscopy while he was a postdoc at UCSF in the 90s. Eventually we were able to recruit him to Janelia as a group leader in 2008. And so in the SIM technique, what you do is it's like regular wide field microscope that you would look at cells with, but instead of illuminating the cell uniformly, you put a grating of light on it. And that grating of light actually creates these sort of moire fringes or beat frequencies against the structures inside of the sample. Those beat frequencies are at a lower period, which you can resolve in the microscope even if you can't resolve the fine structure itself. So basically you change the frequency of the information where you can see it in the microscope. You can go into Fourier space, do a reconstruction, and basically improve the resolution of the microscope by a factor of 2 beyond the diffraction limit. So a lot of people at first yawned saying, oh, a factor of 2. In fact, the Nobel committee yawned, and they said, you know, they didn't have SIM share in the prize because they said, well, that is still bound by the prescriptions of the diffraction limit. But you know what? In some ways, this factor of 2 limit to SIM is not just its weakness, it's its strength. Because first, you don't need to have such incredibly high labeling densities to get to that level of resolution. Second, you take many fewer images and fewer measurements to get a SIM image than you do in, say, STED, the other super resolution technique that shared the prize, or in Palm. And you throw orders of magnitude less light at the sample while you're doing this. Therefore, it is the one method which is really compatible with high-speed live imaging, as you can see in these images, and long-duration imaging. So here's the endoplasmic reticulum in a live cell for 1,800 time points in that movie, which is like 2 orders of magnitude higher than you'd be able to do by any other super resolution method. And here we're doing it at high speed, where we're getting 50 frames a second of data out, which shows exactly how ridiculously dynamic organelles in the cell can really be. And yeah, it's only a factor of 2 of spatial resolution, but look at all of the wealth of information you have by having the time aspect in there as well as the decent spatial resolution as well. And again, you can look for long times with limited damage because the intensities are so low. So here are some examples. The bad news is that Motz died of a glioblastoma in 2011. And so basically I ended up inheriting his group, and I wanted to extend that technology and show people that it was good and that it could be applied broadly. So here are some of the examples from that work. So if you've seen here, what's going on here is myosin 2, non-muscle myosin 2, forms these bipolar filaments when the myosins come together. That's this little dumbbell that you see here. And then more and more are recruited, and the dumbbell has more and more monomers in it, and it gets brighter. But boom, you saw that? A new one didn't form elsewhere. It split off from the first one. So that's the new observation, is de novo formation of myosin filaments is possible, but it's far more common for one bipolar filament to act as the template to grow bigger and bigger until, as they found out in this story, the actin cytoskeleton pulls these filaments apart, and then the new smaller ones then grow again and pull apart. So you end up getting this cascading growth of the myosin network that works with the actin to create the forces in the cell. This was with John Hammer's group at NIH. That paper just came out. Another one that came out late last year is we were looking at Urcat cells, which are T-cell analogs that we would plop against a cover slip with antigens to provoke this immunological synapse. And John's people were able to show that formin is what's responsible for bundling together the actin and radial filaments, which then bend around to form these arcs, which then constrain where the cytotoxic granules are going to come out in the T cell. With Arnold Hare in Tobias Meyer's group at Stanford, they were able to show that in collective migration of endothelial cells, the leading cells send out these coherent fingers, which then are engulfed by trailing cells, and then the actin cytoskeleton of the trailing cells gets modeled after these filaments to guide this collective migration of these cells. And in a paper that came out last year with Jennifer's group, we were using live cell sim to show that in the peripheral ER, where there's putatively both tubules and then sheet-like structures, that many of the things that were believed to be sheet-like structures are actually highly dynamic clusters of small tubular-like structures that then come together, blow apart, and so forth. And it's not enough to have just spatial resolution in these things, because it's extremely dynamic. And so again, you have to have both good resolution in space and time, so you don't get sort of a smearing out of what happens when you're taking the data. So if the knock against SIM is that it's limited in resolution to only twice beyond the diffraction limit, how can we go beyond that? How to break this sort of 100 nanometer resolution limit of SIM? Well, the simple and stupid idea, which you can do in total internal reflection, where you only see the bottom of the cell, is just keep increasing the numerical aperture of your lens to go to even higher diffraction limits, so twice beyond that is better. So you can go to 1.7 NA in TURF, at which case your resolution gets pushed down about 82 nanometers with GFP. At this level, a clathrin-coated pit for endocytosis, as it's pulling away from the membrane, because the TURF only looks at the bottom of the pit, they look like rings. So you can actually resolve now the actual sizes of these clathrin-coated pits, and they don't just look like fuzzy balls. And then you can follow from initiation of a pit until its internalization inside of the cell, that whole path. You can then track the lifetimes of these guys from birth to death and correlate the maximum sizes of the pits to their overall lifetime. And then we also did another experiment where we looked at the relationship between actin and clathrin in cells, because there's a lot of differing literature about the role of actin in clathrin-mediated endocytosis. And so what we're able to show is that just prior to endocytosis, actin is recruited about half of the time. When it is, there's a small but statistically significant shortening of the lifetime. We're also able to show that these clathrin patches that you see are actually clusters of individual clathrin-coated pits that spin out individual pits from them. And we also found that there were actin rings inside of the cell that were just about the same size as the clathrin rings. And we thought that, oh, well, they must be co-localized in helping to pinch off the clathrin bud for endocytosis. It turns out that almost none of them are co-localized with the clathrin, and we still don't know exactly what their role is inside of the cell, but in these extra high resolution SIM images, they're extremely common. So what if we want to go to even higher resolution than that? Well, in that case, you need to use the same photo switching tricks like Palm uses and like Stead uses. So this is something Mats was working on when he passed away, and the idea was to use a photo switchable fluorescent protein where you would use a violet light to turn on all of the fluorescent molecules, then you use blue light to turn off all of those molecules except the ones at the nodes of the standing wave where the light is dark. And then you get these little tiny spikes of residual fluorescence that have very high spatial frequency information associated with them that you can scan across the cell and then get even higher harmonics to get higher resolution. So as graduate student Hesper was able to use this to get 50 nanometer resolution. The problem is it took 900 seconds to get one image, and it certainly wasn't going to be the path to glory for live imaging. So I hired my postdoc Dong to explore this, and what we realized is this principle of turning everything on and then saturating everything except at a node, which is the basis of what I just showed, as well as a stimulated emission depletion technique that shared the prize, is really not a good way to do live imaging, because every time you transition the molecules from one state to the next, you risk the possibility of them getting bleached, and also you're throwing a lot of light on that can create damage to the rest of the cell, and it takes time to do this up and down and up and down and up and down. So what we did instead is we patterned the activation instead of uniform to have one standing wave to turn on that, and then read it out with another standing wave of the same period. So instead of therefore having one standing wave that gives us twice beyond the diffraction limit, we have two standing waves take us 3 times. That takes us to 62 nanometer resolution. And as you can see in this movie, as we've scrolled along, you can see now we can then do live imaging. Again, at this point, we can only go 30 frames instead of hundreds of frames, but still we can do live imaging at sub-second acquisition rates. And again, that's just relating you back to these trade-offs between space and time. So we've used that nonlinear SIM technique, in this case, to study the motility and transport of early endosomes in relation to the actin. And we have enough resolution to actually start to see the shapes of these endosomes inside of the cell, and also to study the role of alpha-actinin in binding together actin filaments and filopodia, or these membrane ruffles that you see here. So the one limitation of all this super high resolution SIM I told you about is, like I said, you're looking only at the very bottom of the cell because you're in total internal reflection. So only about 100 nanometers deep, which is not a lot of the cell. and it means you don't have access to a lot of the organelles. Another problem with SIM in general is that the raw images that you distill into the super resolution image are wide field images. And so when any wide field microscope, if your sample gets more than a few microns thick, the out of focus blur completely blocks your ability to see that fine structure from the SIM pattern. So the one way out of that is, instead of going in turf, is to go just outside of turf, where you then have propagating plane waves that are parallel to the surface of the cover slip but penetrate about a micron deep. So even if the sample is hundreds of microns thick, it doesn't matter, because there won't be any out of focus fluorescence, because the illumination is still confined to about a micron. So we used that with Dan Kehart's group at Duke to look at dorsal closure in Drosophila, where midway through embryogenesis these epithelial cells come up and enclose these earlier amniocerosis cells. So here you're seeing these amniocerosis cells, which are known to go through these pulsating contractions and expansions. But now here with the SIM, you have the resolution in space and time to see the single actin filaments inside of these things undergoing these breathing modes here. In this case, what you're seeing is those epithelial cells coming and encroaching on the amniocerosa, and there's filopodia in the amniocerosa, but they do not have this one protein VASP on them, whereas the epithelial cells all have it. So all of that's great. Now we've gone from basically 2D flatland to slightly thicker 2D flatland. But in the end, the only way you're really going to understand the cell is to look at it in its entirety. And so you need real 3D imaging. In fact, because the cell is dynamic, you need 4D imaging. So really, you need to have high resolution across all 4 dimensions simultaneously to really tease out the movies you need to understand the rules by which the game is played inside of cells. Now, the challenge with that is that again, these trade-offs that you face between space and time. But the good news is that the standard tools that people use for live imaging, which is the wide field microscope and the confocal microscope, give you a lot of room for improvement, because both of those in biological specimens are used in an epi configuration where you shine your laser light from above and collect the fluorescence back. And the problem is that you're getting fluorescence everywhere, but the high resolution information is only from the focal plane. And so the other regions are just creating background, bleaching, damage, etc. So one of the most important innovations in microscopy in this century, in my opinion, was when Ernst Stelzer, then at the EMBL, reintroduced this 100-year-old idea of plane illumination, where instead of illuminating from above, you illuminate from the side, and with a cylindrical lens, in this case, to create a sheet of light, which is just coincident with the focal plane. In that case, you don't have any background, any bleaching, any damage from the regions above and below, because they're dark. Furthermore, the entire plane is illuminated at once. You don't need to scan a point around like in a confocal microscope. You snap an image with a fast camera and you go plane by plane through the specimen. So this has been transformative for studying embryogenesis at single cell resolution. But it has one weakness, which is that there's a trade-off between how flat that light sheet is over a given field of view you want to study and how thick it has to be. And it's great for single cell resolution, but what if, like me, you want to look at single cells but look at subcellular structure inside of them? Well, a cultured cell might be 6 microns thick, but the light sheet necessary to cover that single cell might be 3 microns thick, so then you're not getting much benefit from the plane illumination. So it begs the question, is there a way of decoupling how big the field of view is from how thick the light sheet is? And there is, and it's a physics trick, which is called a Bessel beam. So instead of illuminating the objective, which creates your sphere of light with a circle, you create it with a ring, and the thickness of the ring and the diameter of the ring give you 2 independent knobs to control the length and the width of the pencil of light that comes out. This is exactly the same technology that you go when you check out at the supermarket and the little laser is going zip, zip, zip over the barcode. They're using a Bessel beam because it's non-diffracting and long and thin. and stays small as it goes across that barcode. And so what we then do is we scan that Bessel beam in and out of the screen here while we integrate on a camera, then go to the next plane, next, and get our image. That sounds slow, but in our modern embodiments, we go up to 1,000 planes a second through the specimen, or several volumes per second. This is an example of doing that, where we've actually combined this technique with stepping the beam to be able to do structured illumination microscopy like Mott's developed, but by actually stepping a beam instead of creating a grading pattern directly. And so this is a cell that's been transfected with CSARC, a cancer signaling protein, that promotes these membrane ruffles that fold over and encapsulate the extracellular fluid into these big vacuoles that you see here in this X-ray view of the cell. But now we're studying cells for hundreds of time points in 3D at high speed, and this is really where I wanted to be. Because when I realized the opportunity presented by light sheet microscopy, I really wanted, at that time, it was when I was at my greatest level of frustration with Palm, and the realization that I had to do this live imaging in 3D, and that you know, we needed to do this in a non-toxic and fast way. And so there was an opportunity saying, you know, basically what I learned is, from Mott's, what we learned is that by going away from this monomaniacal focus on spatial resolution to backing off a bit, he had a much greater space to work in than the rest of us, because he had more of these other metrics that were of interest to biologists. And so what I was thinking, well, what if we pull all the way back to the diffraction limit? Could we have an even bigger space in some other axis? In other words, could I make a microscope that would be as transformative in terms of its improvements in speed and low damage for live cell imaging as Palm was transformative in the spatial resolution? And that was really the thought processes that then led to this Bessel beam microscope. In order to make this microscope as fast as it was, though, we had to not just step a single beam. We created an array of 7 beams so that each would only have to go over 1/7 of the field of view. And that was when I discovered probably what was the most, to me, the most shocking thing in my career, was that how much less toxic it was to image with 7 Bessel beams rather than 1. And what I've learned now in doing almost 10 years of live imaging is that while the total dose of light you throw on a cell is an important metric for its health, a far more important metric is the instantaneous power that you deliver into the cell. So seen in that light, the confocal microscope, which is the gold standard of live imaging, may be the absolute stupidest possible way in the world to do live imaging. Because not only do you have these cones of light above and below the focus that are creating all this unnecessary bleaching and damage, at the focus itself you have this actinic spot of light that's going, and it's leaving death and destruction in its wake as it goes through the cell. And so the moral of the story is a line is better than a point, and a plane is better than a line, so why the hell would you stop at 7 Bessel beams? So I started to model what would happen when the Bessel beams got so packed together that they started to interfere with one another. And what I discovered is you go through these periods of resonance and anti-resonance between the side lobes, but there are certain magical periods of the pattern of Bessel beams where 3 great things happen. First, you get near destructive interference of the side lobes of the Bessel beam, which normally create out-of-focus light you don't want. Second, you have 100% modulation of the intensity in the plane, which is ideal for doing structured illumination. And third, you spread the energy out across the entire plane to minimize the damage. So I am an engineer. I am not a physicist. I am certainly not a chemist, and I'm not a biologist. And engineers always think in terms of trade-offs, so you don't get something for nothing. So when 3 great things happen at the same time, that's like a unicorn. And what do you do with a unicorn? You feed it carrots and you take it home, okay? So this was a great unicorn, all right? So this became what became known as the lattice light sheet microscope. So you can run this thing in 2 modes. First, because the pattern is structured, you can move it in small fractions of the period, use Motz's methods of structured illumination, and get higher resolution. Or you can actually dither this pattern back and forth to smear it out. Then you only need to take one image per plane, and then you can image fast. So there's two modes, the high resolution mode, the high speed mode. You can see this is 7.5 times faster. This is about 1.5 times better resolution. So you gain a little bit of resolution versus a lot more speed. Now, it pains me to say this as somebody who has spent 40 years doing super resolution microscopy, but in about 95% of the cases, people would rather have the speed than the resolution. Okay? So an example of using this was with Geraldine Sadhu's group at Hopkins, where they were interested in these P granules inside of the early C. elegans embryo, these liquid-like droplets of RNA and protein. And it was believed that the protein, this MEG protein, would be homogeneous inside of there. But we had just enough resolution with the SIM mode of the lattice to show that there's definitely, it's not homogeneous, but actually have these ribbon-like scaffolds inside of them. But really, the real winner for the lattice was not the SIM mode, like I said, but this dithered mode, where we can go very fast and non-invasively through cells. And so here are some examples of that. Here you're looking at a neutrophil cell, but now you're not looking at motility on a cover slip anymore. You're looking at 3D motility inside of a collagen mesh, again for hundreds of time points. Here's tetrahymena, a pond protozoan, trapped in a pocket of agarose like a jail cell, and he's madly twerking his way to find some way out of there. But for over 1,200 time points at 3 volumes per second. So it blows away any kind of spinning disk microscope, if you're familiar with that. This is looking at Dictystelia, a slime mold amoeboid, and you can see that little lightning bolt there. So when we were taking that data and rendering the movie, we go to our collaborator and say, or the collaborator said to us, what the hell was that? And we said, we don't know, you're the biologist, you tell us. Everybody goes away with 10 terabytes and a big smile on their face with this microscope. And then 2 weeks later, they call us up crying because they have no idea how to deal with 10 terabytes of data. And that is the Achilles heel of this microscope. And that is the biggest pressing need, in my opinion, right now in cell biology, is to have people trained in both, the computational methods and and understanding of the biological questions to extract quantitative meaning from these types of data sets. Because it's easy to get data, but the thing you learn about biology, it's variable, and you have to have high N to find trends in all of that stuff. And if you're doing that in 4D data already with high N, it's a big data problem that has not been solved. If you like these images, Google lattice light sheet Vimeo, and you'll have fun seeing about 40 different examples of how beautiful the cell really is. So one of the things that this turned out to be good for, to my chagrin, was back to single molecules and super resolution again. I actually made a bet when I was at my lowest point about Palm in 2008 with a couple friends saying, I will never do single molecule and super resolution again. I bet them $1,000, and thanks to Lattice, I lost that bet. Because the nice thing about the Lattice light sheet is it's so thin that it's thinner than the depth of focus of the high NA objective you're using to look at stuff. So one of the Achilles heels of doing single molecule experiments in live systems is, again, if the sample, like in SIM, is more than a few microns thick, there's so much out of focus light you can't see single molecules. But with the Lattice light sheet, this is a 35 micron diameter spheroid of mouse embryonic stem cells. You park the light sheet at one plane, and it's like having a 3D version of TURF, where you get great signal to noise for single molecule tracking. This is what you would see with a standard wide field microscope, and that's what you get with the Lattice light sheet. So that's been used to good effect by the Transcription Imaging Consortium at Janelia, where they were able to look at the binding of transcription factors to DNA, and by looking to look at the lifetime of the residence of these binding events, they were able to strip out specific versus non-specific binding and show that transcription forms these hotspots inside of the nucleus. The following year, they were able to show that these hotspots are spatially segregated from heterochromatin in the nucleus. But it also provided an opportunity to revisit single molecule localization microscopy, because like I said, the challenge with Palm and any form of super resolution is getting enough damn label inside of there densely decorate the features of interest. Well, in the same year that we published our Palm paper, Robin Hochstrasser's group at Penn published a related idea where you don't photoactivate the molecules. Instead, the molecules are in the media around the cell, whizzing around under Brownian motion so fast that they're hard to see, but when they stick to a target on the cell, then they're a little glowing dot, and then you can localize that. That's called paint. So the advantage of paint is that you have almost an infinite army of molecules constantly whizzing around in solution. So if you're patient enough, you're going to get enough localization events. The disadvantage of paint is that because there's so many fluorescent molecules whizzing around in solution, there's so much out of focus signal that it's hard to do with high enough SNR to see what you want. So lattice, light sheet, and paint is a great combination. As you can see here, you know, the kind of resolution and so forth we can get, even on thick samples like a 20 micron tall cell in cytokinesis there. So some applications of the lattice light sheet that have appeared so far. This is working with Yuko Momori Kiyasui's group in Riken in Japan, where she was studying EB1, a marker at the endpoints of growing microtubules. We studied probably about 500 different cells through all phases of mitosis on each cell. And what we were able to find is that there's actually the growth. She could tell by both the trajectories and locations of certain EB1 tracks that there's de novo nucleation of new microtubules right at the metaphase plate, right during metaphase in these cells. With Tommy Kirkhausen's group at Harvard Med, we again looked at endocytosis, but now not in turf, so we were limited at the bottom, but at the whole cell and the dorsal surface as well. And what Tommy was able to show is that on the dorsal surface at the leading edge of these migrating cells, endocytosis is largely shut down. And what he hypothesizes is if exocytosis is still happening, you create an imbalance. And so you get a net deposition of membrane to the surface in that case, and then that's what allows you to create these membrane ruffles that you get at the leading edge. With Jennifer again and Gillian Griffiths at Cambridge, we looked at not an artificial system with a Urcat cell on a cover slip, but a real primary T cell hooking up to a dendritic cell and looking at the formation of the immunological synapse and identifying this very fast flow of actin which happens along the sides of the T cell after the establishment of the synapse. Again, with Tommy's group and his great postdoc Gokul, Gokul calibrated the lattice scope they had for single molecule sensitivity to understand the relative contributions of clathrin-dependent and clathrin-independent means of endocytosis of Shiga toxin into the cell. And what they discovered is that while the clathrin-independent means is more common, the cargoes are so much bigger in the clathrin-dependent means that still 85% of the total Shiga is delivered through clathrin. And in a recent study with Jennifer's group, she wanted to do 5D imaging, so they labeled 6 different organelles at the same time in the same cell to do, again, the lattice light sheet live on this, and then did a very exhaustive computational analysis of this to do what she calls unraveling the organelle interactome, to see where one organelle contacts another inside of the cell, how often these contacts exist, how much they persist, and so forth. So the lattice light sheet is not just restricted to doing single cell imaging. We can start to do multicellular imaging. So this is this dorsal closure that I talked about earlier with the grazing incident sim. We don't have the resolution that we had there, but now we can look through the whole amnioserosa here at this process happening. This is looking at a C. elegans embryo just prior to hatching in the threefold stage. Then it's moving like a bat out of hell, and it's too fast to get the whole volume. But we can park at one plane, take 50 frames a second here, and again take 10,000 frames to watch that guy moving around. Here you're watching in the zebrafish in the olfactory epithelium as these neural crest stem cells differentiate and then migrate and then eventually plug into the olfactory epithelium here. But one of the things you'll notice about these images is that we move to this multicellular environment. the image quality starts to degrade more and more. And so that's a basic challenge we face if we want to go deeper with any form of optical microscopy inside of multicellular specimens, but specifically for light sheet, the light sheet going in gets scrambled, and so these meiotic chromosomes look fine here, but it's all dim and blurred out by the time because the light sheet is bloomed and scattered through here. Likewise, on the detection side, even if the light sheet is good, these histones that are closest to the objective are fine, but the histones here have their light has to pass through that embryo and then they get scrambled, and so that's why they're dark and fuzzy there. So it's just like on a rainy day when you have light on your windshield and then causes a poor view. So there are two ways of getting around this problem. The first is to eliminate the aberrations. And so over the last 5 years or so, people have been working on a lot of clearing technologies which homogenize the refractive index in fixed tissue so that you end up getting, a sample which looks like glass with fluorescence in it that you can image. Well, one of the most intriguing clearing protocols is something called expansion microscopy developed by Ed Boyden's group. And so what they do is they infuse the tissue with a polymer gel, and then they cross-link that gel, and they furthermore attach the fluorophores that have already decorated your sample in there to the gel. So now they're doubly linked, first to their proteins, and now they're linked to the gel. And now they put in proteases to blow away all the biological crap. So now what you have left is just a phantom of fluorescence and gel, and then they change from the salt concentration to pure water, and the gel expands just like when a baby pees in its diaper, and the thing gets 5x bigger, and now you have this ghost that's 5x bigger, and then you image that. So they had this paper that came out in Science in 2015, and I saw that paper and I thought it was full of shit. And they called us in January last year and said they'd like to do lattice light sheet on it, and I said, yeah, sure, come on by. And I was sure that I was just going to rub their nose into that shit, okay? And then when they came, the first image we got, this is the soma of a single neuron, and you could have knocked me down with a feather. I just could not possibly believe. So this is a cytosolic fill of this neuron, but all of those little holes you see are the mitochondria that exist because the thing is excluded. You can see all this ribbony stuff. That's the endoplasmic reticulum. You can see the nuclear membrane. You can see hints of the microtubules. It's like a bad quality but still pretty damn interesting TEM image that you're doing a cut through the soma of a neuron in a tissue. But it's done incredibly fast, and it's at an effective resolution because of the expansion of 50 by 50 by 80 nanometers. Do you know how hard you'd have to work by STED, PALM, SIM to get that level of resolution? And you're normally doing it over a field of view like this. So, I thought it was just complete bullshit, but in the end, it's like, holy crap, look what this thing can do. It was amazing to me. So we started to get very interested in working with Ed's guys, and we've been doing that for the last year. So I have to say that, expansion works great in mouse brains. It works great in fly brains. It works like shit on single cells in my experience. If you have any collagen or any kind of connective tissue, all bets are off. So it's got lots and lots of problems, but when the stars align, it's pretty amazing. So here we're doing 3-color imaging with Thi1 YFP for a subset of neurons there. If you zoom in, you can see every spine neck and every spine along these dendrites. You can see every axon. You can see every bouton. Here's a little MIP here where now it's 3-color with antibodies for Homer and Bassoon for pre- and postsynaptic. You can see every goddamn spine have the postsynaptic marker. You can see the presynaptic marker. You can see the synaptic cleft. It's all there. It's all there. And it's just, sometimes the nutty ideas are good ideas, and this is a pretty good nutty idea in the end. And so the thing is, it's great to combine with the lattice because you expand the tissue even 4.5 times. You're talking 90 times the volume. So you have a lot of stuff to go through, so you need to go through it fast. You need to do it in a way that it doesn't bleach the surrounding regions while you're looking over one field of view. And you need something that has higher axial resolution so you have isotropic resolution. So lattice light sheet's the perfect marriage again to this expansion microscopy. So here's some examples in the fly brain. At the top, this is one color channel. The other was looking at all the synapses in NC82, and you can resolve every synapse. But in this case, we cover the entire brain in 60 hours. So 6 teravoxels in each color, 16 bits, 24 terabytes a day in 60 hours to cover the whole brain. This is a cutout from right here. That's the ellipsoid body, the compass of the fly brain. This is our best confocal images from our fly light pipeline at Janelia, and this is what you get of the ellipsoid body with the lattice exam. So it's pretty damn amazing. So when it comes to at least sparse connectomics, where again you have a whole pile of different things with sparse expression patterns, this could be the cat's meow. This could be a really big deal. But one thing, I'm probably, a Nobel laureate when he says something's never going to happen is treading on very thin ice. But I think I'm pretty safe when I say nobody's going to be doing expansion microscopy on live tissue anytime soon. Okay, so what are we going to do about this problem of the aberrations and the scattering when we want to look at living things? Because remember I said, damn it, you have to look at the live cell. Well, the good news is that this was a problem that was solved by astronomers 50 years ago when they developed adaptive optics, where you shine a light into the stratosphere, excite sodium atoms to make an artificial star that's next to the very dim astronomical object you want to see. The light from both of those things comes into your telescope. They've been distorted in exactly the same way by the atmosphere because they followed exactly the same path through the atmosphere. And then you bounce that off a mirror, pick off the light from the bright guide star, put it in a sensor that determines the distortion of the wavefronts, put it in a computer, change the shape of a mirror to exactly balance that, get a flat wavefront and a perfect focus. So bing, you go from that to that. So without adaptive optics, the best ground-based telescope isn't any better than a reflector that you could have in your backyard. But with adaptive optics, the best ground-based telescopes are better in the infrared than the Hubble Space Telescope. So we can play the exact same trick in microscopy by using a 2-photon laser to create a guide star of fluorescence inside of the specimen. Everything else is pretty much the same, except there's one little wrinkle, which is that in this case we're looking at some fluorescent beads that are underneath a zebrafish embryo. With no AO, you can see that all the beads are pretty well messed up. If we do what I just described and we just park the 2-photon laser at a point, like on one bead, we can correct at that one bead, but we still don't have a good correction elsewhere because the aberration is different in those different areas. But what we do instead is we actually, instead of parking it, which also requires some kind of intelligence in the computer to say where to steer this thing, we just scan it over a whole field of interest to get sort of an average correction. And then the fluorescence is de-scanned back off of the galvanometer mirrors to create a point of light which goes in the detector. So you kind of get the average correction. So that, of course, then gives you a better correction over that field than the point correction. But what was surprising is it also was a better correction at the point where you presumably did the best you can with the single spot. And that's because in biological tissue, the aberrations are far more complicated than they are in astronomy. And so what happens is the sensor gets confused by the high Zernike coefficient, basically gets speckles on the sensor that confuses the crap out of them and you get an inaccurate correction. But if you average those out, you lose the very highest modes, but you actually get an overall better correction. That was pretty technical, but and anyway, it works well. So this is looking in the spinal cord region of a developing zebrafish. This is without the adaptive optics, and this is with. So again, you go from low signal and low resolution to high signal and high resolution. And to show them that you can do this over a bigger volume, here we're doing a large fraction of the zebrafish brain, a subset of neurons. As you go from spot to spot in the sample with this 2-photon point scanning microscope, you're going through different tissues, so you need a different correction. So in this case, we had to correct 20,000 different times over little patches to be able to get full resolution everywhere. But we do that correction much faster than the imaging itself in this confocal microscope. So this is turning off the adaptive optics, and that's what you would see with a state-of-the-art 2-photon microscope today. and this is with the adaptive optics. These are mitochondria in the soma of a neuron, 150 microns deep. You turn on the AO. Now, because you know the point spread function, you can deconvolve, and you see those mitochondria as good as if it were a cell on a cover slip. But in this case, this is point scanning confocal, this is point scanning 2-photon. That volume took 18 hours to generate. So not exactly what I want for live cell imaging. So the goal of our group in the last couple of years has been to combine the lattice light sheet technology that we have with the adaptive optics to take the lattice into multicellular specimens. As a result, it's a complicated beast, and it's basically 3 microscopes in one. The lattice light sheet in the center, that's this. Then one adaptive optic microscope that corrects the light sheet itself for the aberrations it faces as it comes in sideways into the specimen. And another adaptive optic microscope that corrects for the light that comes out and is detected. So it's 2 AO microscopes and a lattice microscope. So now this is looking in a developing zebrafish embryo as we go down in the back of the fish, down into the spinal cord region. These are the muscle cells from the side. You can see as we turn on and off the AO how things get blurrier as you go deeper, but we're able to maintain correction right down into the notochord. Then we can build up a volume there, and then we see all these cells here. We can do whatever cutaways we want. Now the difference from before is that this is not that much smaller a volume than I showed you in the last thing. That took 18 hours. This takes 7.5 minutes because it's done with lattice light sheet. And furthermore, then we can study the dynamics of the development for over 75 time points here. So now we're getting kind of this semi-omniscient, no sparrow shall fall kind of understanding of what's happening inside of the organism. So we've done a lot of things with that with Dave Drubin's group at Berkeley. We've looked at endocytosis inside of organoids. These organoids are in a matrigel, and that matrigel causes tremendous aberrations. So you see, I mean, this is the post case, but if you look at this Dynamin channel in particular, when this movie scrolls back around, you can see almost none of these pits. But then with the AO, you can see this is, it looks pretty crappy there. But by the time you turn on the AO, everything is bing. And then here's an example of, with Tommy again, of doing this in an actual zebrafish in a series of muscle cells. And what we find is that you get clathrin-coated pits in the T-tubules that actually span across these muscle cells. Most of them are trapped, but some undergo active transport in between. You wouldn't have a prayer of seeing this without the AO. So now we can also use this to study cell motility. In this case, actually the wiring up of a neural circuit. This is with no AO and with AO in the orthoslices. You build up the volume. You can't see any of these neurites without the AO in any detail. But then as we go through this little guy, this is the spinal cord of the developing fish with a subset of neurons labeled. And then now we can then, finish off. Okay, and then you can see these little guys. These are the little kind of growth cones at the end of these neurites as these little guys are developing and working along. So then you can study again in subcellular detail all the little filipodi on the end of these guys as they're going along. And so then we're able to then look at the dynamics of organelles in the fish. Okay, so this is where I really want to be because again, for me as a physicist, I'm speaking out of order as not being a biologist, but we're taught on our mother's knee about the observer effect, that whenever you look at a system, you perturb a system. And in my opinion, we have never seen the cell in its native state for at least 3 different reasons. First, until recently, as powerful as fluorescent proteins are, we've used them by transfection methods that are pretty uncontrolled and often overexpressed, and that changes the balance of everything else that's going on in the cell. So CRISPR-Cas9 to get to endogenous levels is a great step in the right direction. The second problem is we've been studying these overexpressed cells with confocal microscopes that stress the shit out of the cells even if it doesn't directly kill them. And so we're not seeing the native state for that reason, and the lattice light sheet is a good step forward in that direction. But the final problem is that much of what optical microscopy has contributed to our understanding of cell biology has been by looking at immortalized cells on cover slips, and it's damn for sure that they didn't evolve there. And so we know that the dynamic and structural phenotypes that we observe in our microscope are the result of gene expression, which in turn is controlled by the local environmental cues. So if the environment isn't native, the end result is not likely to be native. So we have to study cell biology with subcellular resolution inside the parent organism where these cells came from. And that's where you need the adaptive optics. So we need to bring all of those pieces together. So that's what we're trying to do here. So here you're looking at Golgi and mitochondria inside of, in this case, in the spinal cord region. This is another example of that in the volume rendering here. Here you're looking at mitochondria by a dye, but then again the Golgi and the ER in this case. And what you find is all sorts of different morphologies for these, particularly the Golgi, that's very different inside of different cell types inside of the organism, and certainly different from the stereotypical Golgi type of thing that you see in cultured cells. And so there's really huge differences in both the dynamics and the location. You can see that these guys all orient to the midline of the fish, all of the Golgi to one side. So there's all sorts of things going on that you could never deduce by looking at a cell on a cover slip. And in my favorite example here, this is now turning on the AO. This is the ear of a developing zebrafish, skin cells, hindbrain, and now these are little dextran particles, and this neutrophil is coming up and scooping up all of these things. So I showed you that movie earlier of a neutrophil in a collagen mesh to say, now we're studying 3D motility. But that's still pretty damn artificial. This is the real deal. I keep saying that everything we've been studying about cell motility of cells on cover slips is like watching lions in a zoo, you know, but this is looking at a lion on the savannah. You know, you're actually seeing it in its real native environment. And so, to me, this is the future of cell biology, to really be able to take the full panoply of tools that the cell biologists have developed over the years for imaging and applying them inside of a multicellular context at high resolution in space and time. And so now I'm ready to close. Just a couple of things. So we've had about 70 different groups come to our lab to do experiments over the last 5 years. And that's been very effective, but it's also too small of a bottleneck for all the people who want to come. And so at Janelia, we've developed an advanced imaging center, which has clones of these microscopes. It doesn't have the lattice AO yet, but will by the end of the year. And everybody can come. It's only for outside people. It's not Janelia people. It's for you guys. So if you have a problem that one of these microscopes you think could address, you can apply. It's only a 2-page proposal. We're not big on red tape at Janelia. And if you're accepted, you come for a couple weeks. You pay for the plane ticket, but we pay for everything else. So, you know, we'll feed you in the pub. You can have drinks in the evening. We put you up in the hotel on site. And, you know, you can send your cells in advance. Hell, you give us time. We will clone whatever plasmids you need or whatever, you know. We'll house your mice in advance or your flies or your fish, have them all staged and ready to go and transfected for the day you show up, and you'll just hit the ground running and take data. People have loved this thing. It's been a roaring success. And the papers are just now finally starting to trickle out of the AC, but that trickle will become a flood. And I highly suggest that if you're interested in these tools, even before you have to worry about building or buying your own, assuming you could, you can at least try it out and get your own 10 terabytes of data to cry over eventually, okay? And so with that, I'd like to close. I mean, there's so many people I have to thank. You know, I've been at Janelia since before the doors opened, when it was a mud pit. And it's been a great ride. But two things. First, the visitor program there has been a secret weapon for us. You know, I really thought it would be isolating to be at a place that was just for systems and neuroscience, but we've been able to pull the best people in cell developmental neurobiology to come work with us there because it's so easy to bring people in there. And the other thing is Janelia really subscribes to the model that I love and that I learned at Bell Labs, which is that less is more when it comes to groups. My group has averaged 3 people at any one point over those 10 years. And I contend that we have output produced groups of 30 to 50, many groups of 30 to 50. And it's because our group is so small and tight-knit, and we work by collaboration with other groups. And I just really think that, for me at least, that has been a far more effective model than the standard model that you typically find in academia. Thank you for your time. Shy just like yesterday. But you always have to have one to break the ice. Getting it? You want to turn up the lights over there too? Yeah, so there's a question in the back. I can't see you, but except for your arm, yeah.
I really like the analogy with watching football to figure out the rules of football. But the watching football is like looking at the bright field. You see everything.
Right.
Whereas the fluorescence, you're limited by the number of the fluorescent molecules.
Right,
As
yes.
you know that the protein works in complex, never individually. So
yes.
how do you overcome that? Because I imagine there's hundreds of molecules inside the cells
Right.
working together.
Right.
But we're limited by the number of fluorescent.
Yeah, so I keep saying there's still two holy grails that have not been touched on well in microscopy for biology. One is label-free imaging, because the label itself is perturbative. And the other is the ability to see dozens or hundreds of proteins at the same time doing everything that we've seen here with one or two proteins, right? And so how we get there is still unknown. We actually, I didn't talk about it, but we have a project. It won't be live, but we have a project where we're doing super resolution imaging and correlative focused ion beam milling scanning electron microscopy at 6 degrees Kelvin. And the point of doing that is the line widths for the fluorophores collapse and get extremely narrow. So there's an opportunity potentially in at least cryo-frozen cells to basically be able to see, hopefully dozens or more proteins, if we can find the chemistry by which we can link them to different things with different fluors. At least the line widths would be narrower, and then correlate that with electron microscopy to get the global contrast that EM provides, along with the protein-specific contrast from that. So there's many, many pieces missing from that puzzle still, but at least that's a path we're going on as a really kind of pie-in-the-sky exploratory direction to try to get to where you want to be. Sure.
There's all these cool imaging technologies. How we can make sure we have this cool technology, microscopy techniques from the bench of physicists,
right,
the benches of neurobiologists.
Yeah, and again,
the imaging quarantine may not be enough.
Yeah, oh, absolutely. It's also too narrow a bottleneck, and part of that is as I go to Berkeley, the first thing I'm going to do is replicate that imaging center at Berkeley. So that's only going to be an hour away for you guys to come and use that. But even that's too little. And so we take three-prong approach. One is things like the AIC. The second is that we document our microscopes incredibly thoroughly, right down to the screws, and we give you all the mechanical drawings, all the control code. We have videos online about how to take it apart, put it back together, align it, use it. Hours and hours of video on all of this. And so if you execute an NDA, we do that, but you have to have somebody who has the chops of an experimental physicist who can put that together with you before you use it. And then the final thing, and the only real solution to the problem, is it has to be commercialized. And in order to be commercialized, that's a long process. You basically you have to get over the valley of death from the initial prototype to a turnkey instrument. And part of that is you have to convince the companies that it's worth the millions of dollars of investment to make that thing. And that's really another reason for something like the AIC, is to build up a demand so that the companies know that it's worth the risk to take. So on the lattice, Zeiss is working on a product now, and hopefully in 18 months they'll have a lattice on the market, which you can buy a lattice now from a company called 3i, but you're going to have to be as astute as a good physicist or microscopist to run it. But eventually the hope is that there is going to be the turnkey box in 18 months that you will be able to buy and be able to do this stuff, hopefully at a price that will allow it to displace spinning disk confocal microscopy. Yeah. Run.
I'm very interested in the lattice sheet microscopy.
Yeah.
So I wonder, what is the wavelength range on both sides? Have you tried? And also, is photo blinking ever become an issue for the images you took?
Yeah, so first on the wavelength range, that's just dependent on what lasers and filters you decide to use, but the one in the AIC has 7 lasers on it from 405 to 647 and most everything in between. So you have a broad range there. And in terms of the bleaching always exists. The bleaching, here, let me see if I got this movie somewhere buried in here. I'm going to compare for you. Yeah, here it is. Okay. So this is on a slide I didn't show, but I'll show you a little movie going on there. Okay. This is Clathrin-coated pits again. The top is looking at it with a spinning disk, and the bottom is looking at it with the lattice light sheet. So you can see the rates of bleaching in the two cases as this goes along. So that's really what you're talking about versus your spinning disk in terms of how much less bleaching you're going to get. It's still there, but you'll go a lot farther.
Are there areas of chromophore development that you think would be really crucial or potentially transformative?
Well, I think there has been a great advance in again, not in an expressed one, but my colleague at Janelia, Luke Lavis, has developed these Janelia floor dyes, JF dyes, which put Alexa dyes or Atto dyes or anything else to shame in terms of their brightness and photostability, and there's about 12 different versions that span from 400 to the near IR. And they're cell permeable and non-toxic and easily deliverable to targets with halo tag. And they are transforming hundreds of labs. He's given out 1,000 free aliquots of this stuff to people already, and they keep coming. It's a really big advance there. Now, anything that could be done in an express probe that would be better would be something that would be greatly received by the community. But for me, an even bigger challenge, and this touches on the question we had first, is what we need is more orthogonal attachment strategies of good chromophores or other things that are expressed to be able to see more things at the same time in the cell. And that's been a real bottleneck. And there hasn't been enough effort in that direction, I think.
Those hardware being packed into a, say, endoscope, so you can actually go in and look at a live animal?
In theory, yes. In practice, that's quite an engineering challenge. It's possible. I could see with the right miniaturization and things like Bragg gratings and other things that one could make something like that. But yeah, that's far in the future. Yeah.
So if you want to do multicolor imaging with the light sheet, do you have to do that sequentially to avoid interference with the vessel beams?
Yes, so exactly, so we do. So right now, that's why we have to use such a fast SLM. So we use a ferroelectric SLM to write that pattern, because we do plane by plane sequential. So we do, red, green, blue, red, green, blue, red, green, blue. And because we're so fast with the camera, we're much faster generally than things are moving in the cell, and it's still easy enough to register with doing that. But I would like to be able to do that, and because, for example, like on the expansion microscopy, so doing that sequential thing in 3 colors like I showed with Homer, bassoon, and YFP, that's fine, but it is 3 times as long. What I would like is a long Stokes shift dye that I could put on one antibody while I have the short Stokes shift dye on the other, and then I could excite both with the same laser, but I can use a dichroic then to split the emission. Then I could use 2 cameras and double my rate of acquisition. I mean, there are ways that you could do it in parallel by clever optics tricks, but we haven't gone down that path yet.
In acknowledgement slide, you listed many people. Could you tell how many of them fit into 3 major categories: hardware, software, and biologists?
Yeah, well, I mean, you know, if I go back to my group slide, okay, this is the biggest my group ever got, okay. So basically, what I have is I have one guy who's totally dedicated to instrumentation code, and he's been the brains of every microscope we've done since 2007. All the other guys are basically, they build the microscopes. They're not really biologists. All of these people over here are the biologists, okay, and that's part of the 70 different groups that come. So basically, when we come up with a new tool, we're well known enough now that we have sort of a core group of biologists that we've worked at again and again on every different tool. We go to them first and say, hey, it's at the bleeding edge now, you want to give it a shot. They try it, we work out the wrinkles and iterate, and as it gets better, then we toot our horns more, and then we try to get other biologists to kind of come in. But we really focus on the nuts and bolts part of it, so yeah. Yeah.
How accessible you think expansion microscopy would be, like for implementing here with our neuroscience group?
For what?
Is it as simple as like ordering the cross-linking reagents?
In principle, yes. Yeah, we had a workshop on EXM and lattice last summer, and so we had, again, about 40 different groups come, and they brought their tissue, and all of Ed's guys came down and they expanded it with their, with Ed's guys' help, and then we put in the lattice-scopes, and it was, I'd say, a very mixed success, okay? I think what you're going to find is that each sample is going to have certain peculiarities, and there's lots of things we look at that just don't work at all. But for the good news is that if you're interested in like mouse cortex or fly, those tissues are pretty homogeneous, and so they expand really shockingly well. I mean, really shockingly. Now, what will be interesting is you can see with the antibodies, the Homer bassoon looks pretty good. What I would like to, we've done stuff at higher expansion ratios to try to get better resolution, and some of that you can see that things are all distorted to hell, okay, that there's still issues there. So I think it's a quickly developing thing, and it's not quite clear where it will have niches, but it's like, man, it's just like everything I've ever done in my career. There's, first there's the disbelief, and then there's everybody getting on and everybody racing ahead without doing any controls and creating a bunch of shit, and then eventually there's the disillusionment phase, and then eventually it asymptotes to some level of reality, right? So we're still kind of on this sort of skyrocketing part of the thing. We got to figure out where that asymptote really is, okay? Yeah.
Hi Eric, thanks for another great talk. I wanted to go back to bleaching, if you wouldn't mind, briefly. I was just wondering if you'd looked into any of the techniques from like atomic physics where they used to depopulate certain energy states. So I guess a lot of bleaching originates from
shelving in a triplet. Right.
And so you can depopulate those optically, or you can interrupt the spin orbit coupling with magnetic.
Yeah, actually Stefan Hell did something like that. He called T-Rex back in like 2006. And it was somewhat effective, but the problem is it made everything really slow because of the dark state lifetime was so long you had to kind of stick on there for a long time to depopulate it out of there. Another thing that my wife Na and I did early on was something we called the pulse splitter. So for damage in 2-photon microscopy, again, you can look at the whole literature on photobleaching, and it's all over the place because it's so difficult to get a uniform control on what's going on. But one of the themes that was constant was that essentially higher power was creating faster bleaching, okay? And so what we did is we basically took, for 2-photon microscopy, where you're getting these pulses at 80 megahertz out of the laser, we created this sort of thing that split the beam into many beams and then popped them back all into the same spot. So basically turned your 80 megahertz laser with pulses of a certain energy into a gigahertz laser of pulses that are much lower in energy. And even though it required root n of the number of pulses higher average power to get the same signal, the damage was actually much lower, much higher than, you know, like factors of 5. And also not just the bleaching, but also like blebbing of cells and stuff like that. It's again, that was kind of also in my mind when I started getting into the multiple Bessel beams, right, is that, you know, peak power is really the killer from both a damage perspective and a bleaching perspective. And that's why you could see on those pits why things persist as long. So, so yeah, there's all sorts of, it's not a simple Jablonski diagram and that's it. There's all sorts of shit going on and it's very difficult to tease out. But empirically, keeping the peak power down is the key.
Thanks.
In terms of the diversity of samples you've looked at, how about the green part of the planet? Have you tried plant samples?
Yes, we did one round with Elliot Meyerowitz's people from Caltech, where they wanted to look at both actin and microtubules, and it worked quite fine. Yeah, we did the lattice AO on that, and I would say that was pretty much a success. There's a lot of background signal from the chlorophyll, but if you're in a big enough different bandwidth, it can work pretty well. Yeah.
All right, I know you guys have a lot of questions, but you know, maybe you can email Eric. We just have to make sure we move on to the next event. So let's give another warm applause for this.